Class 12, welcome back. Hope you're all fine. So today I resume the lesson 2 of your biology book, Sexual Reproduction in Flowering Plants. The topic that we'll be doing today will be outbreeding devices. We've studied that pollination is of two types, self and cross. Now both of these have their own advantages and disadvantages. While in self, plant needs no elaborate preparations for a pollinating agent, the risks are lesser and the chances of seed formation is more. But there is a major problem. No genetic variabilities are produced, which means a huge loss in the race of evolution. No new characters are produced. Just like if we don't learn anything new, we lose out on chances of progress. If an organism does not develop new characters, it will not evolve and then it is running the risk of extinction. But the entire point of reproduction is survival of species. Hence, genetic variability is very important. In order to ensure genetic variability, cross-pollination is preferred by the plant. Even if it means making elaborate arrangements for pollinating agents, developing bright petals, fragrance, producing nectar, etc., and most important, producing huge number of pollens. It's all worth the effort if in the long term plant survives and becomes better adapted to its environment. But there is a problem. Most flowers you come across are bisexual or hermaphrodite. So chances of self-pollination are high. But plant doesn't want that. What do you do when you are asked to do something that you don't want to do? You make excuses. You dilly-dally. That's what plant does. It makes arrangements that even if pollen and stigma are being produced on the same flower, its own pollen is rejected by its stigma. How does that happen is our learning goal for today. So these are our learning goals to discuss about outbreeding devices, find out about pollen-pistil interaction, and let's also introduce ourselves to artificial hybridization. Beginning with our first topic, outbreeding devices. Outbreeding is production of offspring by mating of genetically unrelated individuals. This means fertilization of male and female gametes from two different plants. Why this is important? To produce variations by making new genetic combinations and to avoid inbreeding depression. Inbreeding depression means loss of vigor or decrease in good qualities due to continuous self-pollination or continuous inbreeding. So unless pollen and ovule are from different plants, no variations can be produced. Now, even if plant has bisexual flowers, outbreeding can be managed by using certain techniques known as outbreeding devices. The first strategy is careful timing. You recall the first condition of self-pollination? Pollen and ovule must mature at the same time. What if their maturation is not synchronized? What does this mean? Let's take the first situation. In a bisexual flower, pollen is mature, released for pollination, but the ovule of the same flower is not yet ready to receive the male gamete. Even if the pollen lands on its own stigma, it will be rejected. So no self-pollination. By the time ovule is ready, pollen would already be released, anther would be dried up. So now ovule can receive pollen from flowers of other plant. This strategy is known as proto-andry. Proto means first, andry means male. So this prevents self-pollination. Now take the reverse situation. If ovule is ready to receive pollen but its own pollen are not released yet, It can be pollinated by nearby flowers of different plant. This would ensure cross-pollination. This condition in which ovule matures first is known as protogyny. Can you find out how this term is derived? This type of separation through protogyny or protoandry is temporal separation. Now the second strategy, to understand this strategy, see this photo of primrose flower. Can you see the difference between the two pictures? In thrum, the stamens are much taller and their pollen are picked by insects at almost same point, which matches with the level of stigma of pin flowers. Now, level of stigma in pin flowers is such that its pollen cannot reach its own stigma but can be picked by insects again at a level which matches with stigma of thrum flowers. Now these thrum and pin flowers are the two type of flowers produced by primrose plants in two different plants. One plant will produce only thrum type of flowers, one plant will produce only pin type of flower to ensure physical separation. Can you imagine a situation where thrum and pin flowers are produced on the same plant? This is your first assignment. Find out if outbreeding will happen if thrum and pin flowers are produced on the same plant. Now the third strategy is self-incompatibility. In this, pollen of same flower is rejected by its own stigma. This is based on pollen-stigma interaction, where stigma proteins inhibit pollen germination if the pollen belongs to its own flower. So this is a genetic mechanism. Now besides the above three strategies, there is the fourth method of keeping sexes separate. These methods we have already covered. Now, landing of pollen on stigma ends pollination. But does that guarantee fertilization? Not until pollen is accepted by the stigma. Some of the reasons why pollen would be rejected we just covered. But what if a major error occurs? What if the pollen is of a different species? If these pollen are allowed to germinate, fertilization will fail. So stigma rejects such pollen. It has the ability to recognize the pollen, and this is done by an interaction or conversation between pollen and stigma. This conversation is not verbal but chemical. This chemical interaction between pollen and stigma is known as pollen-pistil interaction. Now let's discuss an experiment where we want to study pollen tube formation. We want to see pollen tube formation. For this, we'll take a drop of 10% sugar solution on a slide. Now we'll take a pollen of our choice. Generally we take pollens of vinca, crotalaria or balsam because these pollens give beautiful images. We dust these pollens on our glass slide and leave for 15 to 30 minutes. The slides we usually take for study of pollen tube formation are known as cavity slides. These are slides which have a small cavity on them or a depression on them. Now after 15 to 30 minutes we observe under microscope. We'll see beautiful images of pollen tubes coming out of pollen grains. This is something similar to what you'll see. Now let's take our experiment a notch higher. What if we want to be in control of which pollen will land on which stigma? This is what a plant breeder does. He has a pollen of very good variety of plant and another equally good variety plant of same species with some other very desirable character. He wants one plant with both these characters, the desirable character of pollen bearing plant as well as the desirable character of ovule bearing plant. What will he do? Controlled pollination or artificial hybridization. This involves these steps. The first step is emasculation or removing the stamen of plant 2, that is the one with the desirable character 2. Now step 2, sprinkle the pollen from desired plant, that is the first plant, on the stigma of this emasculated flower. Step 3, cover the flower with a butter paper bag to prevent any stray pollen landing on it. This is known as bagging. Finally, step 4 is tagging. You tie tags with identification of plant from which you took the pollen, the date of sprinkling, etc. Now, this is how you do artificial hybridization. You are totally in control of which pollen will land on which ovule. The steps involve emasculation, bagging and tagging. Now, a very important question. What if the flower is unisexual? Do you think these steps will be needed? Let me know in comments. With this, we come to the end of today's topic. These are a few questions for you to practice. Good luck. Bye-bye.